Hello everybody, Hassan here, engineer, MBA, and investor. And in today's Sunday morning video, I want to take a look at ARK Invest article published last month by no other than well-known analyst in the space, Alex Yerman. And of course, she wrote this article with someone else as well, Andrew Kim. I'm not aware of Andrew Kim, but nonetheless, here this article was published just over a month ago. And I was going through this article literally a couple of minutes ago. I said, you know what, this could be a great video for today, because one, I didn't really go over this article when it was first published, and two, I think the best way to learn is to basically read. In my opinion, is either to read or listen. I think other than actually creating content, right? I think for Yerman, and she can maybe attest to that. By her writing this article, she's actually learning more than someone who's actually just to read this article. Because when you're writing things, it forces you to think certain ways that you're not thinking if you're reading things or if you're watching things or listening to things, right? I mean, there's different senses involved when you're writing, right? As opposed to just reading an article, right? But the next best thing, if you're not gonna write an article and basically put all that time in researching, is to read it. So that's what we're gonna do, right? So obviously I'm not gonna do it word by word, but okay, so goes through the history here. Okay, so 1953, discovery of DNA structure. Yep, historical overview. So we take a look at that. What's going on with my mouse? Okay. We take a look at that. Scientific revolution in the species are product of natural selection and evolution. The hypotheses beg questions. How do traits pass on from parents to offspring? Yeah, so the Mendel's experiment, that was with the whole plants things, was able to, I'm just summarizing all of this here. It's basically the whole dominant traits and recessive traits and all the good stuff, For the layman explanation, it's like, how come one parent could have blue eyes, the other parent could have brown eyes, but you end up having brown eyes? It's because brown eyes are dominant traits, right, as opposed to blue eyes. So if anything, if people, blue eyes could go extinct in a very long time because they're not the dominant traits, right? So that's just one basic example, right? That's the example everybody uses, blue eyes versus brown eyes. But it's the same concept for nose size, for hairstyle, hair texture, hair length, balding, all that good stuff, right? So, okay, so scientific genetics, so it all started with Charles Darwin, and we lead to today with the birth of molecular biology with Dr. Emmanuel Charpentier. There's a typo here, there's a missing C, and Dr. Jennifer Doudna. And the core concepts. Cells, okay. Each cell has a nucleus, okay. And the chromosomes, right? And that's where you can find the genes, right? Because the chromosomes include both from parents, right? From the man and woman. Of the parents, right? So you have genes. And that's what we're focusing on when it comes to CRISPR. Okay, so structure of DNA, it's basically H bonds. Yeah, okay, helix style, that's fine. And of course, in humans, RNA is a single strand compound that sends message between DNA and amino acids. To understand this process, please consider this analogy. Imagine a teacher who speaks only English to all her students speak only Spanish. The English-speaking teacher wants her students to line up to a single file in alphabetical order according to the last name. Unfortunately, her Spanish-speaking friend can translate the request. In this example, the English-speaking teacher is analogous to a DNA, and her Spanish-speaking friend RNA, while each is analogous to an amino acid in the entire line of children, the protein. Okay, that's one way of putting it. Sure. So anyways, this is the high-level description, right? What a cell is, what a gene is, with a chromosome in the genes in the chromosome, and so on. So, but here we come with gene editing. Primer. What is the potential of gene editing? That's the age-long question, the million-dollar question, right? And the basic example, if you have AAA, but you identify a person with AAT, so now AAA DNA sequence codes for specific amino acids called phenylalanine, and for the unique person, AAT DNA sequence, and for an amino acid called leucine. So clearly, just by changing one letter, you're basically now changing the whole amino acid, as you should. Right? So it's not just a little detail, it's a big detail, right? So now they're talking about certain disease here, how the only difference is literally is only a letter, right? For some diseases, right? So of course here there's a couple of methods for gene editing. Okay. So meganucleus cut the DNA at a specific site. Note that each meganucleus identifies different sequences and cuts at different sites, making the break of both DNA highly difficult and rare. Okay. So it relies on natural occurring repair process to join the strands together. Yeah. Or insert DNA templates such as broken DNA that can fill new bases. Okay. So this is where you have zinc fingers. And I saw something about zinc fingers yesterday. There's like this company I think there was like this company that just recently came out saying that they're a new company and they're betting all their chips on zinc fingers. I mean, I don't even know why that you would think that, it's like, I'd use that analogy. It's like CDs versus digital, right? CDs were great. I mean, CDs were good. I mean, they were doing something that tapes never could do, not just in the storage wise, but in the performance wise and the easyability. Just think about a big tape, VHS tape. You had 5 of them, and it would stack up like this, right? You could have 500 CDs and still be half of that size of 5 VHS tapes. So just think about it, inventory, the shipping, the ease. But, when digital came about, people moved away from CDs, right? So it's the same exact concept here. Look, zinc fingers had a space, Sangamo Therapeutics, okay, that's fine. But, CRISPR is here to disrupt that, right? People should move away from, just stop wasting your time, in my opinion, right? I've already seen the numbers. We've all seen it, the difference between zinc fingers and CRISPR. Even CRISPR first generation of edits from CRISPR Therapeutics or NTLA and so on are way better than zinc fingers, right? So yeah, I mean, this is where CRISPR comes into play in this article, and of course here they're talking about, how it actually works. And yeah, I mean, high level, there's a sgRNA. The sgRNA is combined with a Cas9 protein. That's why you often hear CRISPR-Cas9, for example. The complex binds with the target DNA, locates the PAM. If successful, the complex cuts at the specific site at both strands, right? Once cut, the step afterward more or less are the same as the techniques mentioned before. So it's a natural process that repairs the strand. Okay. What happens during gene editing? What happens? The DNA is cut. The scientists can relay on the non-homologous end joining. This mechanism relies on your body's natural DNA repair process. Typically works with protruding ends of DNA are compatible with a clean cut and no overhang. It is a much more error-prone, and improper repair can cause chromosome rearrangement or tumor growth. Errors can also occur when the overhanging segments are non-compatible. These present themselves in correct random deletion of base insertion of new ones. And scientists can fill the gap in the DNA break by introducing a DNA template. This process is called homology direct repair, HDR, which Graphite Bio has always promoted this, by the way, this term HDR. They're probably the company that, for whatever reason, they're the company that talks the most about it. which is nothing proprietary. CRISPR appears to be superior genome editing method. So this is actually from their big ideas report. This is literally all you have to see. Compare zinc fingers with talons with CRISPR. And the cost is low, time is in days, technical difficulty is low. I mean, technical difficulty, people could literally do it in the garage and literally happen, right? But I obviously am not promoting that. Please don't do that. Targeting mechanism, okay, multiplex editing, not really hard as opposed to the other ones, which are extremely difficult, if even possible, at a secure rate. It's not just about whether or not it's possible, it's about whether or not it's safe and cost effective, right? There's no point pursuing a certain technology if it's not safe and it's not cost effective in the business world, period. End of story. That's where the line ends. You can't just make exceptions, right? You can't say, oh, it worked for that company for that specific disease. No. The FDA is never going to approve something like this long term for many companies on a scale. It may approve a specific product in a specific disease for a specific company that's trying to tackle that other companies are not doing. But yeah, you don't want to bet your chips on a technology that is not necessarily safe or secure as maybe another technology that's in parallel that's doing exactly the same thing in terms of outcome, which is basically potentially curing the disease. Base editing and prime editing. So base editing, prime editing is where they go next level. CRISPR, now the article sort of alludes that now the CRISPR is really shown to be superior as a technology. We move on to a more superior form of technology under the CRISPR branch. That's where base editing and prime editing comes into play. And yeah, I mean, base editing, Cas9, gRNA, target sequence, you go to the specific letter, and that's it, right? As opposed to cutting the whole strand. So you're just substituting a single DNA base with each pass. Prime editing allows for insertion, deletion, and replacement of multiple bases in one pass. Rather than limiting RNA to its role as an identifier of a correct DNA sequence, prime editing enables RNA to synthesize extra DNA bases on a targeted strand. In other words, prime editing offers all the benefits of base editing and no reliance on HDR or flawed repair mechanism with more flexibility. Relative to traditional CRISPR-Cas9, it offers more specificity. Okay. CRISPR-Cas9 can disrupt genes, unlike prime editing, it cannot correct them. In search of genetic diseases, CRISPR-Cas9 based editing and prime editing have taken center stage. Companies like CRISPR Therapeutics, Caribou, Beam, Prime Medicine are conducting trials to discern. Well, Prime Medicine is really early in this space, even though they're the only one with prime editing. But anyway, how these technologies might eliminate genetic disorders and improve human health. Researchers at the University of Michigan recently published their studies on the use of CRISPR-Cas9 to target brown fat cells in mice in hopes of targeting obesity in humans. Researchers at Harvard University and MIT just developed a method of prime editing called twin PE, which is prime editing, that allows scientists to insert or swap out entire genes of humans. Their lab developed the first way to base edit a mitochondrial. This year, the BBC reported that Great Ormond Street Hospital in UK demonstrated the potential of base editing treating Elisa, who had a T-cell acute lymphoblastic leukemia and went into remission after being treated with trial-based edited CAR-Ts. So we didn't cover this article here, but this actually was really popular this year in the UK from Elisa. That was really, really popular, that article. Maybe we'll have some time to cover that in the next video. But yeah, in conclusion, scientific progress over the past 40 years made gene editing more accurate, efficient, cost-effective. Many questions about gene expression. Okay, so we look at this article and it ends with many exciting opportunities to map out complex interrelationships in the human genome. So the only thing I would say is this article sort of ends with the sense that prime editing is the future and everything else doesn't matter. I would highly advise people to listen to Dr. Liu's episode podcast with ARK Invest, actually, where Liu actually mentions that, it actually depends, right? In some cases, you need a boat to go somewhere. Some cases, you need a car. Some cases, makes more sense to take an airplane, right? If you're going from, for the folks in Toronto, if you're going from downtown to maybe uptown, it makes no sense to take a boat because there's no water in between. There's no point taking an airplane because when it's polluted, it's extremely expensive to do that. And three, I don't even think there's any infrastructure to do that. So it makes sense to take a car, right? And you can introduce a new variable, a bicycle, right? Maybe, in certain shorter distance, a bicycle makes more sense, more environmental friendly, it's better for your health and so on. Same thing for CRISPR editing methods, right? I think some disease makes more sense with CRISPR-Cas9. It gets the job done, it's safe, it's proven, just keep it that way, it's cost effective. Then you have other diseases that are more complex, like when we talk about multiplexing edits, probably don't want to do that with CRISPR-Cas9, the first generation. But then there's other set of diseases, it does make sense, right? So you got to pick and choose, right? So that's the only thing I would say in this article that's probably missing, that analogy. I think that analogy would have been great because they used another analogy earlier in this article explaining about the cells there with RNA. So I would love to have seen that, but nonetheless, a great article from Alex Yerman, analyst Alex Yerman. Great article. I like those articles. I like these articles. I learned a lot, and I think a lot of people will learn a lot from this video, right? So thank you so much for watching, guys. As always, subscribe if you're not, like this video if you found value. Let me know in the comments below, guys, what do you guys think about CRISPR-Cas9, base editing, prime editing? Let me know in the comments below. Let me know what you guys think. Hopefully you're having a beautiful Sunday morning. See you guys in the next video. Have a beautiful Sunday and a beautiful week. Thank you.